Anatomy, nervous system. Today we're going to continue the analysis of the spinal cord from a different perspective. If we could change perspective, we would notice other characteristics other than the surface, which is essentially the difference between grey and white matter. Basically, let's imagine cutting the spinal cord into many thin slices with a knife, that is to say, some cross sections. The grey matter, which is composed of the nearby neurons, glial cells and vessels, is located in the centre of these slices. According to creative people, its shape is similar to a butterfly, while for realists it looks like the letter H. Its inner area is hollowed for almost the entire length of the spinal cord, and it's called ependymal canal or central canal. The cerebrospinal fluid flows precisely through this canal so that the spinal cord can be protected from the potential bumps against bone walls and can receive nourishment and other chemical substances from blood. The white matter instead is located on the periphery and is composed of neuronal axons. Neuronal axons are grouped in bundles of nerve fibers sheathed with myelin, from which its typical white look. That's why it's called white matter. The arrangement in the space of these two sections of the spinal cord is unique. The inner part of the spinal cord is perfectly symmetrical. It is composed of two halves, clearly visible by drawing a line between the anterior median fissure and the posterior median sulcus. Each half is called antemere and has a particular arrangement in the grey matter, which forms two horns, one anteriorly and one posteriorly. At the thoracic and sacral segments, there is a lateral horn, which is placed between the two horns made of grey matter and the lateral column made of white matter. The anterior horn has a quadrangular shape. In the most ventral part, bundles of fibers called roots coming out of the spinal cord at the anterolateral sulcus emerge from the ventral part and converge to give rise to the anterior root of the spinal cord. The posterior horn has a longer and thinner shape than the anterior. In fact, it almost reaches the posterior surface of the spinal cord. The apex of the horn, heading laterally and outward, is separated from the surrounding white matter by a layer of nerve fibers called the Lissauer's marginal zone. The dorsal part of the posterior horn receives the nerve fibers from the posterior roots, which enter the spinal cord through the posterior lateral sulcus. The two symmetrical halves of grey matter within the spinal cord are connected by the grey commissure. In the area in front of the grey commissure, we find a layer of myelinated fibers called the anterior white commissure, which separates it from the bottom of the anterior median fissure. Instead, in the dorsal part of the spinal cord, the grey commissure is connected to the posterior medium sulcus through a septum of glial cells called the posterior medium septum. At the centre of the grey commissure lies the ependymal canal, a residual of the canal that extends into the spinal portion of the neural tube, containing cerebrospinal fluid. The name of this canal comes from the epithelium that lines its walls, a ciliated epithelium called ependyma, which gives rise to outward extensions. The ependymal canal continues upwards at the level of one of the cavities within the brain, known as the fourth ventricle, while downward it reaches the spinal cone. Sometimes, at this level, it can be enlarged, hence the name of terminal ventricle. From this point on, the canal gets progressively thinner until it reaches a dead end at the level of the filum terminal. Around the canal, the grey matter appears translucent and granular and takes the name of central gelatinous substance, a layer consisting of glial cells, nerve fibers and neurons. Generally, the ependymal canal in adults ends its obliteration at the age of 70 approximately due to the infiltration of a central gelatinous substance. When examining a cross-section of the spinal cord under a microscope, we can observe a number of other details. Starting from the grey matter, it is composed of cell bodies of multipolar neurons. The classic isoform neurons with a single axon and a dense dendritic network that detaches from the cell body. Based on their morphology and the types of cells they form synapses with, we can speak of type 1 Golgi cells, type 2 Golgi cells. Let's focus on type 1 Golgi cells. Type 1 Golgi cells can be classified into radicular neurons and funicular neurons, based on the direction of their axons. Radicular neurons, as their name suggests, are directed towards the anterior roots of the spinal nerves. At this point, they can be divided into visceral motor neurons, belonging to the autonomic nervous system, and somatic motor neurons belonging to the somatic nervous system. Visceral efferent neurons are mostly located in the lateral horn and are characterized by an axon wrapped in myelin sheath, which reaches the ganglia of both sympathetic and parasympathetic chains. Therefore, they take the name of preganglionic neurons. These neurons receive stimuli from visceral sensory neurons and process a response, forming both direct and indirect reflex arcs. Somatic motor neurons are instead located in the anterior horn of the grey matter, and their axons terminate on striated muscles. We know two types of them. Alpha motor neurons, these are the largest and they innervate muscle fibers of work. Gamba motor neurons, smaller than the former, they innervate neuromuscular spindles, a complex of receptors within the muscle itself. Basically, their function is to assess muscle stretch during contraction. These neurons are connected to somatic sensory neurons, forming reflex arcs, and to higher centers through descending bundles of fibers which make up the motor pathways. Then there are the funicular neurons. Funicular neurons are mostly located on the posterior horn and the grey commissure. Their name comes from the fact that once they leave the grey matter, the axon travels through the white matter in systems of nerve fibers known as bundles or funiculi. Funicular neurons receive inputs from spinal ganglion neurons or other funicular neurons and never leave the neural axis. This means that they do not directly reach peripheral organs, but only form synapses with other neurons. We know two types of funicular neurons. Association neurons. After a more or less extended journey in the white matter, these neurons return to the grey matter in order to create synapses between different segments of the spinal cord. Projection neurons. These neurons have an axon that sends or projects impulses to distant central structures, such as the brainstem. Now let's move to the type 2 Golgi cells. Unlike the previous type of neurons, they have axons that never leave the grey matter of the segment where they are located. Their role is to distribute peripheral and central impulses. Therefore, they are known as short-distance association neurons. Neurons in the grey matter can form bundles where they are all connected to the same nervous elements. These bundles are called nuclei, if spherical or ovoid in shape, or columns if longitudinally extended. Neurons in the anterior horns cluster into motor columns, consisting of motor neurons responsible for muscle innervation and arranged longitudinally. Specifically, there are three groups. The medial longitudinal motor group extends throughout the length of the spinal cord and innervates the muscles of the trunk. There is a specific correspondence between muscles of different areas of the body and specific areas of the central nervous system, CNS. This correspondence is defined as somatotopic organization, which allows the innervation of flexor muscles by neurons located ventrally, while extensor muscles are innervated by neurons located dorsally. The lateral longitudinal motor group is located at the cervical and lumbar enlarged regions of the spinal cord and innervates the muscles of both the upper and lower limbs. The phrenic nucleus, located centrally between the C3 and C7 segments, innervates the diaphragm. Neurons in the posterior horns form four groups. Substantia gelatinosa of Rolando, which, as mentioned before, envelopes the apex of the horns. This is composed by type II cells and larger neurons and extends throughout the spinal cord. Nucleus proprius of the posterior horn, a small cluster anterior to the previous one, also extends throughout the entire spinal cord. Marginal zone, located posterior to the substantia gelatinosa, consists of larger sized neurons. Thoracic and dorsal nucleus, also known as Clark's column, located at the base of the posterior horns, extends between the C8 and L4 segments. Then we have the neurons of the lateral horn, as we said before, extend from T1 to L3, serving as visceral efferent neurons. Altogether, they form an intermediate lateral group and an intermediate medial group. Axons of the lateral group enter into the anterior roots of the spinal nerves and reach the sympathetic trunks, while the neurons of the intermediate medial group serve to link nervous structures, so these are associated neurons. The arrangement of neurons in the grey matter is really unique. In fact, neurons are organized in vertical laminas, placed one next to the other, just like stacked sheets, for the entire length of the spinal cord. Inside each antemere, we can see 9 succeeding laminas, starting from the head of the posterior horn plus the central 10th. First lamina matches the apex of the posterior horn between grey and white matter. Second lamina coincides with the substantia gelatinosa of Rolando. It represents the head of the posterior horn. Third lamina. Its composition is smaller to the second lamina, but is thicker and neurons are bigger. Just like the second lamina, it is part of the head of the posterior horn. Fourth lamina. Just as the second and third lamina, it is part of the head of the posterior horn. It is composed of funicular neurons arranged in myelin fiber bundles. Fifth lamina. It is composed of smaller fibers on the medial side, while it contains the reticular nucleus on the lateral side, built at a cervical level. Sixth lamina. It takes up the posterior horn base, but is well developed only at the level of cervical and lumbar bulges. At the sacral nerve instead, it is composed of efferent neurons of the parasympathetic system, which innervate the pelvis innards. Seventh lamina. It encompasses the intermediate mass and part of the posterior horn base. It contains Clark's column nucleus, which gives rise to one of the spinal cerebellar tracts connecting the spinal cord and the cerebellum, as well as other intermediate neurons. Additionally, it contains funicular association neurons. Along with the sixth lamina, it contains linking bundles to the thalamus. Eighth lamina. It occupies the entire anterior horn at the thoracic level, while it narrows medially at the level of the bulges. The neurons formed in this lamina are funicular neurons, which assume a T shape to connect with the other motor neurons, with the arms of the T directed superiorly or inferiorly. The defining characteristic of this lamina is its dense network of connections, making it a crucial associative center. Ninth lamina. It is situated at the head of the anterior horn and contains motor neurons. As previously mentioned, there is a somatotopic organization regarding the innervation operated by these motor neurons, and this lamina is precisely where motor columns can be identified. 10th lamina. It corresponds to the central gelatinous substance and the anterior and posterior grey commissures that surround the central canal. It contains small funicular neurons. Now let's move to the peripheral part of the spinal cord, the white matter. It is composed of bundles of myelinated nerve fibers, along with longitudinal non-myelated fibers, neuroglia, and blood vessels. As observed in the external structure of the spinal cord, the white matter is divided by sulci and the fissure into anterior, lateral, and posterior columns. The anterior columns on both sides are connected by a sheet of white matter called the anterior white commissure. In contrast, the inner portions of the posterior columns are united by the corner-commissural zone, which is located behind the grey commissure. The myelinated fibers in the grey matter consist of the axons of Golgi type 1 neurons from the grey matter. This forms two types of extensions: funicular fibers from funicular neurons and radicular fibers from radicular neurons. The funicular fibers gather into bundles known as tracts, which can be categorized into two types: projection tracts and association tracts. Projection tracts are composed solely of ascending or descending fibers, while association tracts contain both ascending and descending fibers. The projection tracts include 1. Ascending projection tracts. These originate from neurons in the grey matter and ganglia, which are clusters of neurons located alongside or in front of the spinal column, as well as near internal organs. Their function is to carry sensory information from the ganglia to the spinal cord. These will later be referred to as pathways of general sensitivity. 2. Descending projection tracts. These connect supraspinal centers located above the spinal cord to the grey matter of the spinal cord. Some of these tracts have a motor function, influencing both internal organs and the skeletal muscle system. These are known as pyramidal pathways. The other portion has a regulatory role on motor neurons, known as extrapyramidal pathways. Instead, association tracts link various segments, neuromeres, of the spinal cord internally, running alongside the grey matter. Radicular fibers take the crosswise path and create two fiber systems, one anterior and one posterior. The anterior radicular fibers form the anterior roots of the spinal nerves, constituting the motor roots. Some of these fibers end on the skeletal striated muscles, representing somatic motor fibers. Others originate from the preganglionic neurons of the seventh lamina and end on neurons in the ganglia of the autonomic nervous system, primarily at the visceral level. Hence, these are known as visceral motor fibers. Likewise, the posterior radicular fibers form the posterior roots of the spinal nerves, thus being sensory in function. Upon entering the spinal root, they split in a T-shaped pattern. The ascending branch travels upwards within the posterior column of the white matter until it reaches the medulla. The descending branch, after taking a variable path, concludes within the posterior columns. Certain fibers from both branches can directly terminate within the anterior horn, forming significant reflex arcs for skeletal or smooth muscle.
Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.